All right, hello, my name is Sean Furey, and this is how researchers discovered that tyrosine kinase activity was present in CML patients, starting from research done on chickens. I'm going to give a brief summary because this can be a little hard to follow. So we'll start with how oncogene capture works. So oncogene capture is the process by which a virus can steal a portion of a host cell's DNA and then transmit that to other hosts. So a virus, even a non-cancer causing virus, can infect a host, take a portion of its DNA, and that portion of the DNA can then become cancer causing. And that virus then will have that DNA, and that can be transmitted to other hosts. So the same DNA derived from one particular organism or one individual can be transmitted to other individuals, and it can be cancer causing. That's known as oncogene capture. And in terms of CML patients, well, and just in terms of viruses, there were a couple viruses that were researched, and the cancer causing portion of these viruses was found to be a tyrosine kinase. So there's two viruses really in particular, and those were the Rous sarcoma virus, as well as the Maloney virus, which is also known as the Abelson virus, well, even though they're not the same thing. And then lastly, the tyrosine kinase is found normally in humans, at least this particular tyrosine kinase that can be found in CML patients. However, in CML patients, the tyrosine kinase is mutated and it's translocated to a different chromosome, and that leads to heightened tyrosine kinase activity, leading to cell proliferation and eventually cancer and death. So how did they start? All started with chickens. In 1909, Peter Roos removed a cancerous tumor from a hen. That hen later died. The surgery was not a success, but from this tumor, he ground it up and he strained the tumor to extract just the virus. So he used a strain that was small enough to extract just the virus from the tumor. And then he injected this virus into a chicken that did not have cancer, and he found that the chicken developed tumors. So the chicken without cancer developed cancer from this virus that he had extracted. And this established a relationship between viruses and cancer. So before this, they didn't know viruses could cause cancer, and this was groundbreaking. The virus was named the Rous sarcoma virus, and its abbreviation is RSV. So we're still going to talk about chickens. Really, we're going to be talking about the Rous sarcoma virus. So 50 years later, Howard Temin and Harry Rubin, they create this focus assay, and it allows them to observe the growth of cancer cells, in particular using the Rous sarcoma virus. And they determined that the Rous sarcoma virus has to be an RNA virus, which was unheard of. It was not taken well. And then later, Stephen Martin determines that this gene that the Rous sarcoma virus encodes for that causes cancer is known as SRC. It's SRC. And he determined that by using different temperatures to heighten the activity of the particular genes in the Rous sarcoma virus. And he determined it was SRC that was causing the cancer. And then 6 years later, Bishop and Varmus determined that the SRC gene in the RSV virus came from one particular chicken. So it took them 67 years to determine that the gene, this SRC gene in the Rous sarcoma virus, actually came from this chicken, this super old chicken. And this was one of the first instances where oncogene capture was really researched and discovered. So, yeah. All right. And then this is a little bit back. So we were talking about the 1970s and 1976, and now we're back to the 1970s again. But what happened is some graduate students at Stanford University discovered some enzymes that could enable the joining of DNA. That was called recombinant DNA. Another term for it is called cloning. But basically, it just allowed for research to be developed and move a little quicker. And 7 years later, or a few years later, Erickson and Collette determined that the Sark gene was a kinase. So this Rouse sarcoma virus has a particular gene that's cancer causing, and that's the Sark gene, and they find that that's a kinase. The importance of the recombinant DNA is that Erickson and Collette were using recombinant DNA to conduct their research. 2 years later, Tony Hunter says, "Hey, did you guys know tyrosine kinases exist?" And that was surprising because most kinases are serine and threonine kinases, so it's rare actually to find tyrosine kinases. So here's a quick summary of step 1, because it's a lot. So the Rouse sarcoma virus caused cancer in chickens, and it could be transmitted. And there's a portion of this gene, of the genes in that virus, that came from the chicken itself, and that was oncogene capture. And the gene in particular that we were interested in was the Sark gene. And that was further studied using the cloning method, and they determined it was a tyrosine kinase, which didn't garner much interest at the time. However, it became important later on. Here's step 2, the Abelson virus. So this is the second virus of interest, the second virus that causes cancer and is a tyrosine kinase. All right, so the start of the Abelson virus is in the 1960s. So Herb Abelson, he was doing research on the Maloney virus, which was a cancer-causing virus. He injected it into mice, and they would develop cancer. And the target of that cancer was the thymus, which contains a large number of T cells. So he thought, okay, what if I remove the thymus? Is the virus able to expand its host range? So he removed the thymus using chemical methods. I forget the chemicals he used in particular, but when he did that, he would remove the thymus on completely new mice, and he would re-inject the Maloney virus into these mice so that the virus no longer has the target, the T cells. And what happened is the virus still spread and caused cancer. And its target it changed its target into the B virus, or not the B virus, the B cells instead of the T cells. But Abelson discovered this isn't the same virus. So he injected the Abelson virus into normal mice to see, okay, these mice have a thymus. Is this virus going to target the thymus, or is it going to target the B cells? And if it targeted the B cells, then it's a new virus. And it did. It targeted only the B cells. And it produced the same type of rapid cancer that caused death very quickly. So he called this transformed virus, he called it the Abelson virus. A few years later, 1975, Naomi Rosenberg created a way to watch the Abelson virus replicate and spread cancer in a petri dish. She did this because the Abelson virus was particularly interesting to watch. It spread fast. It was a liquid-based virus, and those made things a lot easier just to research and to watch. So she created this array which allowed her to watch the Abelson virus replicate. And then a year later, Owen Witt uses her development, and he derives the portion of the Abelson virus that encodes the protein that causes cancer. So just like in the Rouse sarcoma virus, they discovered the Sark portion was causing the cancer. They found that in the Abelson virus, there was 2 sets of proteins, and they originally thought it was an unusually large protein, but these 2 proteins were known as GAG-ABL, and those were the proteins that cause cancer in the Abelson virus in comparison to SARC in the Rouse sarcoma virus. And later he found that just like with the SARC virus, or with the SARC protein, GAG-ABL was a protein, or a tyrosine kinase. In particular, ABL was a tyrosine kinase. 3 years later, right? Yeah, 3 years later, 1979, Steve Goff, he's comparing the Maloney virus to the Abelson virus. He's looking at, okay, what is the particular difference in the DNA sequence between the Abelson and the Maloney virus? And what he finds is that the Abelson is missing portions of the Maloney virus, but it also has additional DNA. And what he found is that ABL had actually come from the mice. So the portion of GAG-ABL, the ABL portion of GAG-ABL had come from the mice. And so they knew, okay, this is oncogene capture again. This is just like the Rouse sarcoma virus and the SARC gene coming from the chicken. The ABL came from the mice. All right, so here's a summary of the Abelson virus. And after this, we're going to put it all together a little bit. So the Rouse sarcoma virus and SARC had inspired interest in other viruses. And one of those viruses that was inspired to research was the Maloney virus, which also caused cancer, just like the Rouse sarcoma virus. And they found that when injecting the Maloney virus into different mice, one with thymuses and one without, it actually produced a different virus. This was the Abelson virus. It had a particular protein, which was the ABL protein, which was that ABL protein came from the mouse, so it was oncogene capture. And they also found that the Abelson virus encoded a tyrosine kinase. So you've got two tyrosine kinases. You've got one from the ABL protein, and you've got one from the SARC protein, and you've got oncogene capture in both of these instances. All right, and now let's connect the dots. So this is step 3. So we're going to go back in time and we'll work forward, and it's going to connect the research that we just learned about into the research of the Philadelphia chromosome and CML patients. So 1959, David Hungerford and Peter Knoll, they discovered a mutation in chromosome 22 of CML patients. So what they found was that what they thought was a deletion in chromosome 22. It's just at the end of the chromosome, and they found it in particular in CML patients, and that was later termed the Philadelphia chromosome. 1970, Janet Rowley is like, okay, you're actually wrong. It's not a deletion. What it was is it was a reciprocal translocation between chromosome 9 and 22 in CML patients. So they originally thought there was a deletion in chromosome 22, but what happened is some of 9 and some of 22 had actually switched. And she used a GM sustain with quinacrine mustard, which enabled her to do that. That actually stains the bands of the chromosome, so she saw in particular where the translocation was occurring. And this is the discovery of where it was occurring in particular. So a few years later, 1982, Nora Heisterkamp and John Groffen, they developed a probe. That's not supposed to say prove, it's supposed to say probe. And that sought out ABL, the ABL protein from the Abelson virus in particular. And one of their friends or brothers was Gerard Griswold, and he worked with CML patient tissues. And they knew that CML patients had a particular ABL or particular difference in their chromosomes, and they thought it might be worth trying. So they sent their probe to their brother, Gerald Griswold, and he tested their probe on CML patients, and he found that the ABL is present in normal human chromosomes on chromosome 9. But they also found that in CML patients, only a portion of the ABL is present on chromosome 9, and the rest was on chromosome 22. And that's that translocation. And then a year later, Owen Witt's students, and Owen Witt was the one who isolated GAG-ABL, But one of his students, he's messing around with the GAG-ABL antibody, which is what Witt used to isolate GAG-ABL. And he finds that it's particularly active in cells that came from CML patients. So 1 year apart, they're finding that ABL is present in human cells, and in particular, CML patients. So they're finding ABL's present, and it's different in CML patients. And that difference is where the translocation is. Now here's steps, or the last portion of connecting the dots. So a year later, Nora Heisterkamp and John Groffen, the same ones who were researching the translocation between 9 and 22 using the ABL probe, they sought to find out where or what is combined with ABL in humans, because in mice you have GAG-ABL. It's a combined protein. In humans, what they found is that there's a particular region in CML patients where it would break, or where it would translocate. And they found this region, it was a full protein. It had stopping and ending points in amino acids in its amino acid sequence. And they called this the breakpoint cluster region because it would generally break in this region. And they called that protein the BCR-ABL. So BCR for the breakpoint cluster region and ABL for the Abelson virus. The same year, Ali Kanani, he discovered that the ABL gene in CML patients encodes copious amounts of RNA when compared to normal patients. So to summarize, or to connect that a little bit, so if they know that the ABL gene is encoding for more RNA in CML patients, and they also know that the ABL gene is a tyrosine kinase, then they know there's heightened tyrosine kinase in CML patients, and that's what's causing their cancer. So here's a summary of connecting the dots. So CML patients, they have a mutation between chromosomes 9 and 22. It's a translocation. ABL is normally found in humans on chromosome 9. That doesn't have heightened tyrosine kinase activity. It has its normal required activity. However, BCR-ABL is found on both chromosome 9 and 22 in CML patients, and that's that translocation. And ABL was found to code for copious amounts of RNA in the CML patients compared to normal patients. So here's a full summary. This is the complete breakdown of all the research that was done. So this is steps 1 through 3. So they find Rouse sarcoma virus. It's causing cancer in chickens. There's a particular gene, and it's called the SARC gene. And they know that SARC gene actually came from the chicken itself, just like ABL was in the humans itself. And then they study the SARC gene using recombinant DNA, and they find that it is a tyrosine kinase. And then later, the research in RSV and SARC inspired interest in other viruses, and the Maloney virus gave rise to the Abelson virus, and there was a particular protein called the ABL protein, same thing as the SARC protein. The ABL protein was a tyrosine kinase. It came from the mice, just like the SARC came from the chicken. Both were tyrosine kinases, and both caused cancer. And then they later found, okay, let's connect this to humans. CML patients, they have a mutation. It's a translocation between chromosomes 9 and 22. ABL, the same protein from the Abelson virus. ABL is normally found in humans without cancer on chromosome 9. In CML patients, there's a translocation mutation on chromosome 9 and 22, and BCR-ABL is what that mutation is sort of called. It's those two proteins that are translocated. And they found that this RNA, or this ABL in this translocation, was encoding for copious amounts of RNA compared to normal patients. So in CML patients with this mutation, you're seeing a lot of transcribing from ABL. And they put it all together. They said, okay, we have proteins that are causing cancer. They're tyrosine kinases, and Abel is a tyrosine kinase. It's found in CML patients. It's transcribing in excess, therefore, or in excess or constitutively in the cell, and therefore it's producing excessive amounts of tyrosine kinase. So they discovered, okay, Abel, or the BCR-ABL is what's leading to heightened tyrosine kinase activity in these CML patients. And that's essentially how research on chickens led to heightened tyrosine kinase activity in CML patients. I hope you enjoyed my talk.